Today's topic is inflammation. Inflammation is a complex immune response characterized by redness, heat, swelling, pain, and loss of function. It is a physiological response to a variety of stimuli such as infection and tissue injury. It involves various immune systems and numerous mediators. In general, an acute inflammatory response is rapid and lasts only for a short while. Acute inflammation is accompanied by a systemic reaction known as the acute phase response, which is characterized by a rapid alteration in the level of several plasma proteins. In some diseases, persistent immune activation can result in chronic inflammation which lasts for a long time. The inflammatory process. In the early stages of an inflammatory response, the predominant cell type infiltrating the tissue is the neutrophils. Neutrophil infiltration into the tissue peaks within the first 6 hours of an inflammatory response. At this time point, the production of neutrophils in the bone marrow increases to meet the need. A normal adult produces more than 10^10 neutrophils per day, but during a period of acute inflammation, neutrophil production may increase as much as tenfold. When the neutrophils reaches the tissue, it started expressing increased level of receptors for chemoattractants and consequently exhibit chemotaxis, migrating up a gradient of the chemoattractant. Among the inflammatory mediators that are chemotactic for neutrophils are several chemokines. Complement split products like C3A, C5A, and C5b67, fibrinopeptides, prostaglandins, and leukotrienes. In addition, some of the molecules released by microorganisms like formylmethionine peptides are also chemotactic for neutrophils. Activated neutrophils express increased level of Fc receptors for antibodies and receptors for complement. This enables them to bind more effectively to antibody or complement-coated pathogens and increase phagocytosis. Activation of neutrophils also stimulates metabolic pathways that causes respiratory bursts where reactive oxygen intermediates and reactive nitrogen intermediates are produced. Release of these reactive intermediates and the release of mediators from primary and secondary granules of neutrophils like proteases, phospholipases, elastases, and collagenases plays an important role in killing various pathogens. These substances also contribute to the tissue damage at the inflammatory site. Accumulation of dead cells and microorganisms together with accumulated fluids and various proteins makes up to form pus. Infection or tissue injury induces a complex cascade of non-specific events known as inflammatory response that provides early protection by restricting the tissue damage to the site of infection or tissue injury. The acute inflammatory response involves both localized and systemic responses. Localized inflammatory response. The major characteristics of localized acute inflammatory response first described about 2000 years ago are swelling or tumor, redness or rubor, heat or color, pain or dolor, and loss of function. Within minutes of tissue injury, there is increase in vascular diameter called vasodilation. This results in an increase in the volume of blood in the area and a reduction in the flow of blood. The increased blood volume hits the tissue and causes it to redden. Vascular permeability also increases, leading to leakage of fluid from the blood vessels, particularly at post-capillary venues. This results in an accumulation of fluid, also called edema, in the tissue, which causes swelling and redness in the area. When fluid exudes from the bloodstream, the kinin, clotting, and the fibrinolytic systems are activated. Many of the vascular changes that occurs early in local response are due to direct effect of plasma enzyme mediators such as bradykinin and fibrinopeptides, which induce vasodilation and increase vascular permeability. Some of the vascular changes are due to the indirect effect of the complement anaphylatoxins like C3A, C4A, and C5A, which induce local mast cell degranulation with release of histamine. Histamine is a potent mediator of inflammation, causing vasodilation and smooth muscle contraction. The prostaglandins can also contribute to the vasodilation and increase vascular permeability associated with the acute inflammatory response. Within few hours of the onset of these vascular changes, neutrophils adhere to the endothelial cells and migrate out of the blood into the tissue space. This neutrophil phagocytosis invading pathogens and release mediators that contribute to the inflammatory response. Among the mediators are the macrophage inflammatory protein MIP-1 or chemokines that attract macrophages to the site of inflammation. Macrophages arrive about 5 to 6 hours after an inflammatory response begins. These macrophages are activated cells that exhibit increased phagocytosis and increased release of mediators and cytokines that contribute to the inflammatory response. Activated tissue macrophages secretes three cytokines, interleukin-1, interleukin-6, and TNF-alpha, that induce many of the localized and systemic changes observed in the acute inflammatory response. All three cytokines act locally, inducing coagulation and an increase in vascular permeability. Both TNF-alpha and interleukin-1 induce increased expression of adhesion molecules on vascular endothelial cells. For instance, TNF-alpha stimulates expression of E-selectin and endothelial adhesion molecules that selectively binds adhesion molecules on neutrophils. Interleukin-1 induces increased expression of ICAM-1 and VCAM-1, which bind to integrin on lymphocytes and monocytes. Circulating neutrophils, monocytes, and lymphocytes recognize these adhesion molecules on the wall of the blood vessels, adhere, and then move through the vessel wall into the tissue space. Interleukin-1 and TNF-alpha also acts on macrophages and endothelial cells to induce production of the chemokines that contribute to the influx of neutrophils by increasing their adhesion to vascular endothelial cells and by acting as potent chemotactic factors. In addition, interferon gamma and TNF-alpha activate macrophages and neutrophils and promote phagocytic activity and release of lytic enzymes into the tissue space. The duration and intensity of the local acute inflammatory response must be carefully regulated to control tissue damage and facilitate the tissue repair mechanisms that are necessary for healing. TGF-beta play an important role in limiting the inflammatory response. It also promotes accumulation and proliferation of fibroblasts and the deposition of an extracellular matrix that is required for proper tissue repair. Systemic acute phase response. The local inflammatory response is often accompanied by a systemic response known as the acute phase response. This response is marked by the induction of fever, increased synthesis of hormones such as adrenocorticotropic hormones and hydrocortisones, increased production of white blood cells or leukocytosis, and production of a large number of acute phase proteins in the liver. The increase in the body temperature inhibits the growth of a number of pathogens and appears to enhance the immune response to the pathogen. C-reactive protein is an example of acute phase protein whose serum level increases 1000-fold during an acute phase response. It is composed of 5 identical polypeptides held together by non-covalent interactions. C-reactive proteins binds to a wide variety of microorganisms and activate complement, resulting in deposition of the opsonin C3b on the surface of microorganisms. Phagocytic cells which express C3b receptors can then readily phagocytose the C3b-coated microorganisms. Many systemic acute phase effects are due to the combined action of interleukin-1, TNF-alpha, and interleukin-6. Each of these cytokines acts on the hypothalamus to induce a fever response. Within 12 to 24 hours of the onset of an acute phase inflammatory response, increased level of interleukin-1, TNF-alpha, and interleukin-6 induce production of acute phase protein by hepatocytes. Leukemia inhibiting factors or LIF and oncostatin M or OSM also induce production of acute phase proteins. TNF-alpha also acts on vascular endothelial cells and macrophages to induce secretion of colony-stimulating factors like MCSF, GCSF, and GM-CSF. This CSF stimulates hematopoiesis, resulting in transient increase in the number of white blood cells needed to fight the infection. Chronic inflammatory response. Chronic inflammatory response develops when antigen persists for a long time. Some microorganisms are able to evade clearance by the immune system, for example, by possessing cell wall components that enable them to resist phagocytosis. Such organisms often induce a chronic inflammatory response which results in significant tissue damage. Chronic inflammation also occurs in a number of autoimmune diseases in which self-antigens continually activate T cells. Chronic inflammation also contributes to the tissue damage and wasting associated with many types of cancer. The accumulation and activation of macrophages is the hallmark of chronic inflammation. Cytokines released by the chronically activated macrophages also stimulate fibroblast proliferation and collagen production. A type of scar tissue develops at sites of chronic inflammation by a process called fibrosis, a wound healing reaction that can interfere with normal tissue function. Chronic inflammation may also lead to formation of a granuloma, a tumor-like mass consisting of a central area of activated macrophages surrounded by activated lymphocytes. The center of the granuloma often contains multinucleated giant cells formed by the fusion of activated macrophages. These giant cells typically are surrounded by large modified macrophages that resemble epithelial cells and therefore are called epithelioid cells. Two cytokines in particular, interferon gamma and TNF-alpha, play a central role in the development of chronic inflammation. T helper 1 cells, NK cells, and T cytotoxic cells release interferon gamma, while activated macrophages secretes TNF-alpha. Activation of macrophages by interferon gamma promotes increased transcription of the TNF-alpha gene and increases the stability of the TNF-alpha mRNA. Both effects result in increased TNF-alpha production. TNF alpha acts synergistically with interferon gamma to initiate a chronic inflammatory response. Both cytokines together induce much greater increase in IKM1, E-selectin and class I MSC molecule than either cytokines alone. The increase in intracellular adhesion molecule facilitates the recruitment of large number of cells in a chronic inflammatory response. A region of plump endothelial cells resembling high endothelial venules or HEVs appear along the vasculature in tertiary extralymphoid sites of chronic infection. These HEV-like regions express several mucins that are often displayed on normal HEVs. Several cytokines that are associated with chronic inflammation play a role in the induction of HEV-like regions along the vasculature. Development of this HEV-like vasculature facilitates a large-scale influx of leukocytes which contributes to chronic inflammation. As a conclusion, I would like to say that inflammation is a complex immune response against varieties of stimuli and it involves many cells and mediators. It may be acute response or chronic response. Acute response is rapid and lasts for a short time, while chronic response persists for a long time. Acute response may be localized or systemic. Localized response is restricted to a region of infected tissue, while systemic response is a whole body response. Localized response is often accompanied by systemic response. During inflammation, many different types of cells are recruited to the inflammatory sites, and this is possible with the aid of numerous mediators. Inflammation is also marked by change in the level of various plasma protein. In chronic inflammatory response, the antigen persists for a long time and it often accompanied by massive tissue damage.